Good evening, bachon. Very good evening. Good evening, participants. Actually, what happened? I came by 6:30 in the house. It was some work at the college. I just slept for 5 minutes, and I just woke up with your messages just now. So sorry from my side. Um, I was just tired, and I couldn't know when my eyes just closed out. And I went, okay, but somehow we still have time. For the next 30 minutes, we finish with the yesterday's topic, that is this one, no? Western blotting, all these lectures. This was left. We were done with the chip, pi mole, these topics we did yesterday. So we will not waste our time. We will start just now. So more students will keep joining us then. So MBBT, blotting techniques. So as we have discussed already, they are like southern, northern, western, and you also told me about eastern. I will not discuss that in this part, but there is southern, western, northern, western, dot, and zoo blotting. So we will learn each of them with the technique and some virtual lab regarding the same. So first technique, that is your southern blotting. It is done to detect your DNA fragments by gel electrophoresis, invented in 1975 by E.M. Southern. Just a second, coming back. So in this southern blot, we have a DNA from the samples. We cleave them with the restriction fragments or restriction endonuclease, and then we put them on the gel electrophoresis. And these double-stranded double helix of DNA fragments are denatured according to the pH, and then gel is blotted on the nitrocellulose membrane and transfer the DNA strands to the sheet. Then a probe consisting of purified single-stranded DNA corresponding to specific gene is poured over the sheet. So any fragments that has nucleotide sequence complementing the probe sequence will be hybridized with the probe. So here you can see you have your electrophoretic is performed using a radioactively labeled markers as a size guide in the first lane. Then the gel is covered with a sheet of nitrocellulose placed in a tray of butter on the top of sponge. We will discuss about them, how we transfer your proteins onto the nitrocellulose membrane. Then we slowly remove this nitrocellulose membrane from the gel, and then we put them in a plastic paper and put them with the radioactive labeled nucleic acids. Then you have a molecular markers on the left, the size markers, and on the right hand side you will see radioactive label bands. So information obtained from the southern blotting, we can following information about the technique of southern blotting. So we can understand that how many genes of copies of a genome is present, how much similarity is present between the chromosomal gene and probe sequence, whether rearrangements have occurred during the cloning process or not. We can get to know about paternity testing, sex determination, monoclonal leukemia populations, particular DNA sequence of particular organism, identify gene mutations, deletions, duplications. So all these things are possible with the help of this technique. So now DNA gel electrophoresis. So you can see, keep watching this downside, and it will keep telling us how this. You put your sample first on the gel, Then you run your gel. So this is how you transfer of your bands on the nitrocellulose membrane is done. So, so that was about southern blotting. Now comes the northern blotting. It will be the same, but instead of DNA, we have RNA in this. So principle is almost similar. Everything is similar. You isolate your RNA, you put over the gel, then you transfer to the nylon membrane, and then you hybridize with the buffer. Then you remove the unbound probes. Then you check it with the RQ radiograph on the X-ray film. So you get a particular gene in which tissue is expressed. If it is expressed within a certain stages of development, the quantity of mRNA could be quantified accurately whether the genomic DNA probe has regions that are transcribed. Then use of northern blotting. So northern blotting allows researchers to determine gene expression patterns. This indicates a myriad of practical applications, allowing researchers to compare patterns. We can able to understand unknown proteins, detect the size of RNA, and the various homology. So the principle is same, little bit different. So let's check how it is done. Your northern blotting.